Thank you very much, Tom, and thank you all. I'm here today speaking on behalf of Dr. Mike McGrew. He's a researcher at the Roslin Institute, an animal research institute in Edinburgh, and I work in Mike's group. He would like to have been here himself today, if he could have made it. So Roslin would like to cryopreserve much of their stock of poultry lines that they have. It's a research institute, and we're starting to use semen freezing as a rapid means of collecting and archiving that material. We use the standard method that many of you will be familiar here with horses, cattle, or mammals. Collect the semen, mix it with a freezing mix, load it into straws, freeze it with liquid nitrogen. And to recover a line, we can just take straws, do one round of AI, and inseminate wild-type hens, and we will have back a line of birds that carries a particular gene of interest for those researchers. Usually they're just interested in a particular gene related to the development of the animal. But of course, to recover a rare breed would be a lot more difficult. You would have to do multiple back crosses using the semen straws to inseminate subsequent generations of offspring, and that would take time. Could take 3 years to get back 98% of the genome of genetic material of the original rare breed. But Mike is pioneering the use of another means of preserving material from chickens, and this is using what we call primordial germ cells, which are stem cells that go on to develop in the animal to produce sperm and egg. And these stem cells are present in the laid egg and in the young embryo, and we collect them at 2 days of embryonic development from the bloodstream, and we can expand these cells in culture, freeze them down in a conventional low temperature freezer, we don't need liquid nitrogen, and store them at minus 150 degrees. And thaw them, we can inject them back into a 2-day embryo, and they will migrate to the gonad. They develop into sperm and eggs in the adult sexually mature animal. So that is a way of preserving the genetic material of these animals using these stem cells. The problem is that the host animal that you inject it into also produces its own stem cells and produces its own sperm, so that's eggs. So the donor material is diluted out. If we had a perfect host where the germ cells that produce sperm and eggs in the host animal were suppressed and could provide a niche for the donor rare breed stem cells to proliferate, we would have a way of recovering the complete genetic material of the original rare breed. And we can do that. Mike is using the method of genetic modification to produce birds with that can act as hosts to these donor germ cells. And we have a female line that can be injected with female stem cells. These go on to produce eggs, and we recover material with 100% the identity of the donor in those eggs. If we could extend this, and research is going on to males, we would also be able to produce sperm, let's say, of a rare breed in a male host. We could cross those two hosts, and then within 1 year we would have back a full, authentic breed from these stem cells. So that's the alternative system. We're using both methods at the moment, but the use of these stem cells would be cleaner. It would be technically demanding, so expensive for the collection, but cheaper in the long run because you would more quickly recover your rare breed. And if we could extend this technique to other species, imagine that what we could do, we could protect the genetic material of species that are threatened with extinction in the wild. And that would be the basis of the frozen aviary that Mike has posited and was covered by the BBC earlier in the year. Thank you.